Everybody, welcome back to today our channel is the part of chemistry. Today we will talk about a very general topic like in forensic science most of the peoples are using Ninhydrin test. Ninhydrin test is one of the part of our chemistry topic. So you can see here the caption. The Ninhydrin test mostly used for checking for amines or the alpha amino acids. This is the structure of Ninhydrin. Okay, you can see that what is Ninhydrin test and why the people will use nowadays in forensic science department. What is the purpose and what is the limitation of Ninhydrin test, we will discuss briefly within the 5 minutes. Okay, you can see here the what is Ninhydrin and what is the brief introduction about the Ninhydrin test. You can see the Ninhydrin. The synthesis of Ninhydrin is originally described as 1, 2, 3 triketohydrin. So synthesis of Ninhydrin is mostly it is called as the 1, 2, 3 triketohydrin, but it's also referred to as 1, 2, 3 indene trione. So the first reported by the Professor Human is the one of the great professor in University of Cambridge in England in 1910. He describes about the Ninhydrin test and he brought the very briefly what is Ninhydrin and what are the color changes of Ninhydrin. These things he is bring out to the nowadays people are using forensic science. You can see the marks developed by the Ninhydrin in vary the color from reddish purple to deep purple, being influenced by the composition of mark and the substrate it is deposited on. You can see the here picture easily will understood the various finger tips. You can see that here finger marks are on white copies and paper developed by using the Ninhydrin. You can see, suppose if you touch in your any newspaper and you can touch into that any your things, they will spray these chemicals and then you can find out this your fingerprint matching. That's these techniques they use nowadays in forensic science department and they detect the thieves and some kidnapper peoples. So you can see what is Ninhydrin. You can see here the Ninhydrin test is the chemical test which is used to check whether given the analytes contains amines or the alpha amino acids. This is the main concept of the Ninhydrin test. So Ninhydrin chemicals test is used for determine whether an analyte contains any amines or amino acids. So in this Ninhydrin is chemical formula is C9H6O4. This is the actual Ninhydrin chemical formula. Then you can see the what is the IUPAC name 2,2 dihydroxy indene 1,3 dione. This is added to the test solution of the analytes. The formation of deep blue color within the test sample to indicate that the analytes contain ammonia, primary or secondary amino acids or both. This is the actual mechanism of the Ninhydrin test. Now we can see here what somewhat brief introduction about the Ninhydrin test. Ninhydrin is used to bioanalytical techniques. Ninhydrin is one of the bioanalytical techniques to particularly for the amino acid analysis method. Ninhydrin react with the alpha amino group of primary amino acids produce Reuman's purple. So the chromophores formed in the same for the all primary amino acids. That point is very important. So the intensity of the color formed deep depends on the number of chemical nature of amino groups being analyzed. So for this test, with the primary, secondary and tertiary amines, amino acids and proteins, a deep blue or purple color is observed. So hydroxyproline and proline on reaction with the Ninhydrin, yellow color is formed. So asparagine on reaction with the Ninhydrin produce a brown color. These are the different kinds of amino acids will react with ninhydrin. They will give you a different color. That is the point of Ninhydrin test. Then you can see that ninhydrin react with amino acid in free amino acid groups in the test sample have oxidized the compound, leading to the deelimination. And this reaction, two gases at release. There are the ammonia and another one is the carbon dioxide. So protein bound amino group. So these can be demonstrated by the ninhydrin Schiff's reaction. The neutral pH and 37 degrees Celsius, ninhydrin react with alpha amino group to produce aldehyde, which can then demonstrated by Schiff's reagent. Then you can test. Another one test is how to test for the protein. What is the test for protein? Suppose they will give you a mixture of our compound, then how you will identify the given the analyze or analytics, those analytes, the proteins are present or not. So that is the one of the test is Burette test. Burette test is one of the confirmation for either protein or present or absent. You can find out here the simple procedure. Add 2 ml of sodium hydroxide and 5 to 6 drops of copper sulfate solution to it and shake the test tube gently. Mix the ingredient thoroughly. Allow the mixture to stand 4 to 5 minutes without disturbance. So it appears bluish violet color. It indicates the presence of protein. Suppose if you are giving the mixture of solution and this solution after 4 to 5 minutes by adding of these all like sodium hydroxide and the copper sulfate, then shake well, then after 4 to 5 minutes you can observe in your test tube. Then the test tube, the color will change in bluish and violet color, then indicates your solution is contain protein or protein was present. So the ninhydrin test is not effective to detect the high molecular weight protein, has the steric hindrance limit of the ninhydrin from reaching the alpha amino group. You can see briefly next slide. Then what is actual of the ninhydrin test? You can see here the amino acids of the analytes results in the formation of molecules that is comparable of diketohydrin. The color of this combination is deep blue, that is sometimes referred to as a Raman's color. Actually this is that based on the following mechanism of ninhydrin test. You can see this is that reaction of ninhydrin, what is happened. The amino group of free amino acids is subjected to chemical reaction with ninhydrin and which gives the property of acting as the oxidizing agent. When exposed to the ninhydrin, the amino group undergoes the oxidative deamination, resulting to release the carbon dioxide and ammonia gas. And aldehyde as well as the formation of hydrogenation and the other byproduct, the reaction which is reduced form of ninhydrin. This is that test for ninhydrin. Then what is ninhydrin called? Generally we discussed the previous slide. Ninhydrin is nothing but of 2,2 dihydroxyindane 1,3 dione is one of the organic acid. This is the formula used in the ammonia and detection of ammonia and the amines. Upon reaction of these amines, ninhydrin gets converted to deep or purple derivatives called as Raman's purple. Then what is the wavelength to quantify the amino acids? So this point is very important in various competitive exams. They will ask you. So like ninhydrin react with reaction is the method to detect the quantify amino acids via ninhydrin coloration. So amino acid react with ninhydrin. The reagent detect using the two wavelength. One is the 570 nanometer and another one the 440 nanometers. These are the two wavelength to measure the ninhydrin test, okay, for the quantify of amino acids. Then you can see here the various derivatives of ninhydrin. You can see ninhydrin, 5-methoxy-ninhydrin, benzo-ninhydrin and the 5-methyl-thionine-hydrin. These are the some derivatives of various colors exposed nowadays developed for the ninhydrin test. Then you can see what is actual of this test tube. You can see image, you can understood there is no color, then negative of the amino acid. Suppose yellow color is there, then you can indicate the presence of proline. Then purple color is there, the positive test of amino acids are present. These are the test for the various amino acid presence or not in your given mixture. Then you can see that what is the objective of Ninhydrin test. To determine the main concept of Ninhydrin is to determine the presence of amino acids and amines in the test solutions, to determine the amount of amino acids in the sample, and to distinguish between the amino acid and carbohydrates. These are the objectives of Ninhydrin test. Then what is the limitation? Means you can see that limitation of Ninhydrin test. Ninhydrin test react it not only for the amino group but it also nitrogen, ammonia, other free amines. Because the steric hindrance, earlier we discussed this point, the Ninhydrin test is cannot detect the high molecular weight of protein. It prevents the Ninhydrin from reaching the alpha amino group. This is the limitation of Ninhydrin. Then you can see what are the 4 types of protein structure: primary structure, secondary structure, and quaternary structure, and like tertiary structure. Primary structure, secondary structure, and tertiary structure, and quaternary structure. These are the 4 types of protein structures. Then you can see what is the structure of peptide. You can N-terminal and C-terminal. You can see glycine, serine, valine, and these indicate like the C double bond O and NH. This red color indicate these are the peptide bonds. This is the structure of actual peptide bonds, the between of the jitter ion like amino acids. One amino acid to another one amino acid linkage peptide bonds. And then recently you can see that in newspaper the idea was Ninhydrin test is did for the one of the Andhra Pradesh Maji Minister Vivekananda. He is killed by the someone others. Then people who are nowadays forensics department CBCID, they use for the Ninhydrin test to detect that their fingerprint test for the finding of the people who kills by this Viveka. These are the actual formulas for Ninhydrin test in Telugu they mention here. Really, thanks for your great patience. Like, share, comment, and thank you for your great support. You will meet you next class in some other topics. Thank you very much. Bye.